Genome editing tools in molecular biology introduction. Genome editing is a procedure that allows site-specific modifications in the genome of any organism. The goals for genome manipulation are to learn how new genes function. The second major use of genome manipulation is the creation of alternative treatment options for a variety of genetic disorders. The evolution of genome engineering using designers nucleases has brought a paradigm shift in the field of biotechnology. Talents and CRISPR are crucial in elimination of undesired genes. These tools have demonstrated promising results in various domains, including medical and agricultural sciences. The appealing agricultural applications of nucleus-based genome editing include improved varieties of crops with high yields and desired features like enhanced nutritional content, greater shelf life, better stress tolerance, disease, and pest resistance. The use of ZFN in marine animals such as zebrafish and many livestock animals has been shown, while TALEN and CRISPR systems are used in the cell lines of many cattle species such as cows, pigs, and chickens. Technological advances are crucial for innovative biological research. The real breakthrough in genome engineering came with the design of nucleases that were able to specifically recognize and cleave target DNA. There are three different gene editing nucleases currently available, like ZFNs and TALENs and CRISPR-Cas9. Cas9. So zinc finger nucleases ZFNs. Zinc finger nucleases ZFNs are a class of artificial DNA binding and binding endonucleases that facilitate targeted editing of the genome by creating double-strand breaks in DNA at user-specified locations. So ZFN-induced double-strand breaks are subject to cellular DNA repair processes that lead to both targeted mutagenesis and targeted gene replacement at remarkably high frequencies. Zinc finger nucleases ZFNs are based on zinc finger proteins, a family of naturally occurring transcription factors fused on an Flavobacterium ocenocytes endonuclease. Zinc finger domains can recognize a trinucleotide DNA sequence. A series of linked zinc finger domains recognize longer DNA sequences. FOKL endonuclease works as a dimer, which means that the double-stranded DNA cleavage occurs only at sites of binding of two ZFNs to the opposite DNA strands. The system is based on two ZFNs engineered to recognize different closely located nucleotide sequences within the target site and requires the simultaneous recognition and binding of both the ZFNs, which naturally limits off-target effects. So here ZFN homodimer binding to DNA shown as a 3-finger zinc finger linked to the FN domain through a flexible peptide linker. At the end terminus of each ZFN resides a nuclear localization signal or LLS. The FN domain is linked to the C-terminal finger, finger 3 of the zinc finger domain. For most efficient cleavage, there is no amino acid linker between the zinc finger domain and the FN domain. The binding sites are arranged in an inverted orientation so that one ZFN is making most of its major contacts with one strand of DNA, whereas the other ZFN is making most of its major contacts with the other strand of DNA. So between the two binding sites is a nucleotide spacer NNN, the sequence of which does not seem to be important. When the DNA binding and DNA cleaving domains are fused together, a highly specific pair of genomic scissors are created. ZFN is targeted to cleave a chosen genomic sequence. The cleavage event induced by the ZFN provokes cellular repair processes that in turn mediates efficient modification of the targeted locus. Then mechanism. ZFN is a widely used programmable DNA binding protein derived from the African clawed frog Xenopus levis, ZFNs utilize custom DNA endonucleases to make DSPs. So, ZFNs belong to the naturally occurring family of transcription factors fused with endonuclease FOKI. As a heterodimer, the ZFN subunits contain the ZF domain and FOKI restriction nuclease domain, natural cleaving activity. The FOKL domains dimerize for DNA cleavage and accomplish DSPs on targeted DNA sequences. The DSPs helps the genome editing tool to work. So, mechanism of zinc finger nuclease inducing double-stranded breaks DSPs at the target region in the genome. Two different left and right ZFNs monomers consist of a tandem array of different zinc finger protein recognize a nucleotide triplet shown in blue color. So each ZFN is fused with Flavobacterium ocanocoides. Each ZFN is fused with Flavobacterium ocanocoides. Nuclease and results in dimerization of the FOKI. So DNA cleavage takes place in the 5 to 7 base pair of the spacer sequence shown in red between the ZFP recognition sites. Double-strand breaks are important for site-specific mutagenesis in that they stimulate the cell's natural DNA repair processes, namely homologous recombination and non-homologous enjoining, NHEJ. So these processes are harnessed to generate precisely targeted genomic edits, resulting in cell lines with targeted gene deletions, integrations or modifications, and lead to gene knockout. This approach is used in reverse genetic studies in Drosophila, Melanogaster, zebrafish, rats, and Arabidopsis theriana and mammalian somatic cells. If the break is introduced by homology-based process to introduce recombinant DNA or entire transgenes transferred into the chromosome, referred to as gene correction or gene addiction. This approach has been used to make novel alleles of Indi, Melanogaster, mammalian cell, tobacco, and human embryonic stem and induced pluripotent stem cells. So, application for ZFN, functional genomics, target validation, creation of gene knockout in multiple cell lines, complete knockout of genes not amendable to RNAi, cell-based screening, creation of knock in cell lines with promoters, fusion target, tags or reporters integrated into endogenous genes, and cell line optimization, creation of cell lines that produce higher yields of proteins or antibodies. Then gene therapy, zinc finger nucleases are very powerful gene modification tools. Then transcription activator like effector nucleases. TALEs proteins were first reported in 2009, derived from phytopathogenic bacterial genus Xanthomonas. TALE is a special class of proteins that can bind to DNA and offer flexible applications in genetic engineering due to its compatibility with many functional domains. Different associations of tail proteins with transcriptional activities, repressors, or endonucleases give them potential transformation from transcriptional modulators to genome editing tools are now available. So, transcription activator like effector nucleases are artificial restriction enzymes. Functions as a novel class of genome editing tools that provide precise insertion, deletion, or substitution of specific genes in order to alter the genome. So, TALEs consist of a designed TALE domain that mimics the natural transcription activator like effector proteins and a nuclease that can cleave DNA in cells. They are made by fusing a TAL effector DNA binding domain to a DNA cleavage domain, a nuclease which cuts the DNA strands. So, transcription activator like effectors can be engineered to bind to practically any desired DNA sequence. So, when combined with the nuclease, DNA can be cut at specific locations. So, TALEs are fusion proteins of bacterial TALE protein and FOKL endonucleases. And these restriction enzymes are, introduced into cells. They can be used for gene editing or for genome editing in situ, a technique known as genome editing with engineered nucleases. So TALEs have emerged as alternative to ZFNs for genome editing and introducing targeted DSBs. So TALNEs are similar to ZFNs and comprise a non-specific FOKI nuclease domain fused to a customizable DNA binding domain. This DNA binding domain is composed of highly conserved repeats derived from transcription activator like effectors, TLs, which are proteins secreted by Xanthomonas bacteria to alter transcription of genes in host plant cells. Tail proteins contain three functional domains. Their N-terminal domain harbors the bacterial secretion signal and a non-specific DNA binding activity that is required for the general affinity of the protein to DNA. Their C-terminal domain harbors an interaction interface for the plant transcription factor IAA to functional nuclear localization signals and an acidic activation domain. The N-terminal region of a TALE contains a type of 3 secretion signal, T3SS, and 4 non-canonical repeats, NCR. The C-terminal part, a transcription factor binding site, TFP, 2 nuclear localization signals, NLS, and an acidic activation domain, AAD. Each TALE also harbors a repeat region with a varying number of highly conserved repeats of 33 to 35 AA amino acids placed in tandem. The amino acid sequence of a consensus TALE repeat with 34 amino acids is shown, and the amino acids responsible for the DNA specificity of a tail, the repeat variable diresidues RVD, are highlighted. Also shown are the 5 most commonly used RVDs and the nucleotides they specify. Then TALEN's mechanism. The simple relationship between amino acid sequences and DNA recognition of the TALE binding domain allows for the efficient engineering of proteins. Once the TALEN's constructs have been assembled, they are inserted into plasmids. The target cells are then transfected with plasmids, and the gene products are expressed and enter the nucleus to access the genome. Alternatively, TALEN constructs can be delivered to the cells as mRNAs, which removes the possibility of genomic integration of the TALEN expressing protein. Using an mRNA vector can also dramatically increase the level of homology directed repair or HDR and the success of integration during gene editing. TALEN technology can be used to edit genomes by inducing double-stranded breaks, which cells respond to with repair mechanisms. Non-homologous enjoining reconnects DNA from either side of a double-strand break where there is very little or no sequence overlap for annealing. This repair mechanism induces errors in the genome via insertion or deletion or chromosome rearrangement. Any such errors may render the gene products coded at that location non-functional. Because this activity can vary depending on the species, cell type, target gene, and nuclease used, it should be monitored when designing new systems. DNA can be introduced into a genome through NHEJ in the presence of exogenous double-stranded DNA fragments. Homology directed repair can also introduce 4-8 DNA at the DSB, as the transfected double-stranded sequences are used as templates for the repair enzymes. So, overview of DNA DSB and subsequent repair mechanism. The conventional TALEs require two corresponding DNA recognition sites like TALEN F and TALEN R, flattening with an unspecified central spacer region. The dimeric form of the focal transnuclease domain fused by a single polypeptide shown in brown line is necessary for DSB. The genomic DSB generated by TALENs can be repaired by HR or NHEJ, resulting in the desired genome editing. The NHEJ mediated pathway leads to the generation of a small INDELs. HR with double stranded repair template DNA leads to precise gene editing and gene replacement. And various uses and values of talents. Talents are valuable for the formation of knockout strains and for the study of cell mutations of various organisms like yeast, bacteria, rat embryonic stem cells, fish, xenopus, human cell lines, rodents, and plants. The talents can be formed to enable introduction of mutations into the open reading frames of viruses such as hepatitis B, HIV, and herpes, which are present in the latent state inside the body and are unaffected by treatment that inhibits viruses and their replications. So, hence, their symptoms are permanently evaded with accurate genomic modifications. So, talents allow effective induction of targeted alterations in various model organisms that are challenging for genetic manipulation, like roundworm, fruit fly, zebrafish, pig, rat, and frog. They are also involved in endogenous genomic modification in cells of silkworm, cricket, and cow. So, recent improvements in talents techniques support the study of genomes in wheat plants and allow rapid modifications in the agronomic properties of the crop. They also help in avoiding the need for non host DNA. The improvements in crop properties include increased yield and increased immunity against several diseases. In 2019, physicians at the Great Ormond Street Hospital announced the first clinical use of talent-based genome editing. 11-month-old baby suffering from CD19 plus acute lymphoblastic leukemia was treated with modified donor T cells that had been engineered to attack leukemia cells to be resistant to alimtumab and to evade detection by the host immune system after introduction. A few weeks after therapy, the patient's condition improved. The first genome edited crop to market came in 2019. Next, CRISPR-Cas system. Prokaryotes evolved several defense mechanisms to protect themselves from viral predators. So, clustered regularly interspaced short palindromic repeats and their associated proteins display a prokaryotic adaptive immune system that memorizes previous infections by integrating short sequences of invading genomes, then termed spacers, into the CRISPR locus. The spacers interspaced with the repeats are expressed as small guide CRISPR RNAs that are employed by Cas proteins to target invaders, sequenced specifically upon a reoccurring infection. Many bacterial clustered regularly interspaced short palindromic repeats, CRISPR-Cas associated Cas systems employ the dual RNA associate guided DNA endonuclease Cas9 to defend against invading phages and conjugative plasmids by introducing site-specific double-stranded breaks in target DNA. Target recognition strictly requires the presence of a short protospacer at adjacent motif, flattening the target site, and subsequent R loop formation, and the strand scissions are driven by complementary base pairing between the guide RNA and target DNA, Cas9 DNA interactions, and associated conformational changes. The use of CRISPR-Cas9 as an RNA programmable DNA targeting and editing platform is simplified by a synthetic single guide RNA stRNA, mimicking the natural dual transferring CRISPR-Cas RNA tracer RNA, CRISPR RNA, cRNA structure. In this system, small guide RNAs, cRNAs, are employed for sequence-specific interference with invading nucleic acids. So, CRISPR-Cas comprises a genomic locus called CRISPR that harbors short repetitive elements, repeats, separated by unique sequences or spaces which can originate from mobile genetic elements, such as bacteriophages, transposons, or plasmids. The so-called CRISPR array is preceded by an AT-rich leader sequence and is usually flanked by a set of Cas genes encoding the Cas proteins. So CRISPR-Cas systems can be divided into two main classes, which are further classified into 6 types and several subtypes. The classification is based on the occurrence of effector Cas proteins that convey immunity by cleaving foreign nucleic acids. In class 11, CRISPR-Cas systems type 1, type 1, 3 and 4. The effector molecule consists of a multi-protein complex, whereas class 2 systems types 2, 5 and 6 uses only one effector protein. CRISPR-Cas9 structure. The CRISPR-Cas9 nuclease system require two components: the Cas enzyme for cutting the target sequence and a single guide RNA, sgRNA, which binds to the target sequence of 20 base pair. The target sequence complementary to the sgRNA sequence is followed by two cytosine nucleotides because the sgRNA binds best when the opposite DNA strand is comprised of any nucleotide followed by two guanines. This sequence is called a protospacer adjacent motif sequence. The PAM varies depending on the origin of Cas9. CRISPR-Cas9 nuclease system. CRISPR-Cas9 mechanisms of action. CRISPR-Cas9 creates specific double-stranded breaks at the target locus that trigger DNA repair mechanisms. These corrections result in two types of genome modifications, constitute knockouts through non-homologous engineering and knock-ins through homologous recombination. Then Cas9 mechanism. The key step in editing an organism's genome is selecting targeting of a specific sequence of DNA to biological macromolecules. The Cas9 protein and guide RNA interact to form a complex that can identify target sequences with high selectivity. The Cas9 protein is responsible for locating and cleaving target DNA both in natural and artificial CRISPR-Cas systems. The Cas9 protein has 6 domains: REC1, REC2, bridge helix, PAM interacting, HNH and RUVC. The REC1 domain is the largest and is responsible for binding guide RNA. The role of the REC2 domain is not yet well understood. The arginine rich bridge helix is crucial for initiating cleavage activity upon binding of target DNA. The PAM interacting domain confers PAM specificity and is therefore responsible for initiating binding to target DNA. The HNH and RUVC domains are nuclease domains that cut single-stranded DNA. They are highly homologous to HNH and RUVC domains found in other proteins. Domains are shown. In engineered CRISPR systems, guided RNA is comprised of a single strand of RNA that forms a T-shaped comprised of one tetraloop and 2 or 3 stem loops. The guide RNA is engineered to have a 5' end that is complementary to the target DNA sequence. It forms one tetraloop and 2 or 3 stem loops. Target complementary region is shown in red. The artificial guide RNA binds to the Cas9 protein and upon binding induces a conformational change in the protein. So the conformational change converts the interactive protein into its active form. The mechanism of the conformational change is not completely understood, but Jinek and colleagues hypothesize that steric interactions or weak binding between protein side chains and RNA bases may induce the change